Thanks for joining me for some more Parker Biology. This is our thrilling conclusion of a photosynthesis recap. So we looked at the light reactions. We saw how solar energy was converted into the two forms of chemical energy, ATP and NADPH. And now we're over in the Calvin cycle, in the stroma of the chloroplast. And here we can see three main steps. First, we see carbon dioxide fixation. And carbon dioxide is going to enter the Calvin cycle and join up with an organic compound known as RuBP, ribulose bisphosphate. The enzyme that catalyzes this union is Rubisco. So when carbon dioxide joins up with an organic RuBP, that is carbon fixation. Now a 5-carbon compound plus 1 carbon from CO2 is going to result in a really short-lived 6-carbon intermediate, and it's going to break pretty quickly into 2 3-carbon molecules. And this is why typical plants who use this type of Calvin cycle are referred to as C3 plants, carbon with 3. So the first phase was carbon fixation, and the second main phase is known as reduction. And what that says is that the energy from the light reactions, ATP and NADPH, are going to be used to rearrange the carbons and to ultimately build sugar. The sugar that comes out of the Calvin cycle directly is known as G3P, and this will go through a few more steps and go on to become glucose and other organic compounds. If it just stopped there, eventually there would be no more RuBP, no one to welcome carbon dioxide to the Calvin cycle. And so the third essential step of Calvin is to regenerate the CO2 acceptor. In order to regenerate RuBP, a little extra ATP is spent. It's this extra ATP that explains why cyclic electron flow is necessary. Linear electron flow takes care of these energetic needs, but cyclic compensates for the extra ATP needed to regenerate RuBP. If a plant starts to dry out, if they start to lose too much water, their stomata can actually start to shrivel in and close, and that cuts off gas exchange. The problem with that is oxygen can build up inside their cells, and they won't have enough CO2 to efficiently carry out the Calvin cycle. Also, Rubisco really isn't picky enough, and if there's too much oxygen in the cells, Rubisco will settle for oxygen, try to join oxygen up with RuBP, and the result is a phenomenon known as photorespiration. Photorespiration is very bad. It wastes these organic resources, and it does not result in sugar production. Energy and resources are absolutely wasted. Sugar is not made, and it is very counterproductive. Photorespiration is bad. Some plants, though, are specially adapted to live in dry environments and have a couple of cool possibilities for mechanisms that prevent photorespiration. Now, a plant doesn't get to wake up one day and say, oh, it's really dry outside. I think I'll grow a new adaptation. Not at all. The plant is either born with these specialized modifications or not. So the first type of adaptation to allow some plants to live in really dry, hot environments is known as a C4 adaptation. And C4 plants, instead of just having mesophyll cells, also have a deeper layer of what we call bundle sheath cells. And in C4 plants, carbon fixation and the Calvin cycle occur in 2 separate locations. When CO2 enters, instead of having all the gases intermingling and having Rubisco and RuBP all in the same place, CO2 is quickly taken up by Pep Carboxylase. Pep Carboxylase is going to help escort the CO2 to a deeper tissue layer into the bundle sheath cells, where Calvin cycle will occur. Here, RuBP is kept away from the temptation of any oxygen that might be building up. By keeping this in a separate place, CO2 gets safely to the Calvin cycle. RuBP and Rubisco are not tempted by anyone else, and sugars actually get produced. We often sum up C4 plants then as a spatial separation of carbon fixation and the Calvin cycle. The other option for a cool adaptation is a temporal separation or timing separation, and this occurs in CAM plants. Here, the carbon dioxide is taken up at night. When the stomata are open at night, they don't run the risk of losing so much water from the plant. Organic acids will keep the CO2 safe through the night and then release them to Calvin cycle during the day. So in C4, we see a spatial separation, mesophyll, and Bendel sheath cells, whereas in CAM, we see the timing separation. Bring your CO2 in at night and then use it to build sugar up during the day. Hopefully all of this information has you feeling a lot better about the light reactions in the thylakoids, the Calvin cycle out in the stroma, background information about wavelengths and pigments, action spectrums, absorption spectrums, even redox reactions, and finally the adaptations that some plants utilize to better survive in hot, dry environments. Thanks for joining me for Parker Biology.